Hi, everyone. And then for the next 50-ish minutes, be going through the NWB guide tutorial together, and I'll show you how to use this desktop app to convert data from a variety of different neurophysiology data formats to NWB. We'll get started by going to this website, nwbguide.readthedocs.io. This is the main documentation page for guide. And hopefully you all have, who want to follow along, will have already downloaded guide. Please take this minute now to install guide by opening the executable file and running through the install process. Just to orient you, when you land here at the top, installation instructions, then tutorials. And the third one here is our ecosystem format support. And this shows all the different data types that are supported by NWB for automatic conversion. And you would navigate it through the bottom selector here. So EC-EPHIS stands for extracellular electrophysiology. So this is your extracellular probe in the brain. And there are many acquisition systems out there that record this kind of data, which speaks to the need for a common data standard. And so the name of the format is listed on the left, and whether or not guide supports it is listed on the far right here as a checkbox. So if you have a particular data format, let's say you work with spike GLX data because you are using neuropixels probes. Well, you're in luck. Guide supports that. If you're working with Plexon data in the PLX formats, we support that. OpenEPHIS, we fully support that, etc. You kind of browse around there. If there are formats that you work with that are not supported by guide, I highly encourage you to go to the NWB Guide GitHub page and go to the issues here and click New Issue. And there is a option here for requesting format support, and this will open a little template for you to fill out to request that we add support for a particular format in guide, which is backed by another tool called NeuroConv. Yeah, and I just showed you for extracellular EPHIS, the recording data types. There's also a number of spike sorters that are, many of which are supported by guide, either directly or through spike interface. IC-EPHIS, intracellular electrophysiology is not yet supported. Optical physiology data types such as TIFF files from scan image are supported. Optical physiology types data that comes after segmentation or processing, such as through Kaiman or CNFE, are supported, and some common data types for behaviors such as sleep and deep lab cut are also supported. This would kind of give you an overview of all of that, and please request support for any data types that you are working with that come from an acquisition system or a processing tool that you'd like us to add to NWB Guide. Okay, now moving on to the tutorials. In order to get started with the tutorials, in order to complete the full length of the tutorial, we highly recommend that you register for the DANDY archive, the main archive for storing neurophysiology data supported by the US Brain Initiative. If you haven't registered for DANDY yet, that's okay. You can just follow along on that part of the tutorial, and we can help you later, or you can try the upload process later. Okay, so the first thing we're going to do to run through these tutorials is to generate an example data set. And that is described here in the example dataset generation page. We're going to generate some example extracellular electrophysiology data in the SpikeGLX and Phi formats. And SpikeGLX is a acquisition system that is popular for acquiring neuropixels data, and Phi is a popular tool for doing and managing and curating spike sorting results. So to generate this data set, first we will launch guide. I'm on a Mac. On Windows, it'll be under your applications or programs. Clear this out. And go to the settings. And over here will be a test data set. And I already generated one, so I'm just going to delete that. And I want you to follow along and click generate to generate this test data set. And this will take about a minute, depending on your system. This will generate, I think, a minute or two of fake neuropixels data, and then do a kind of rough spike sorting of that data and processing to get that into the Phi format. And we will be converting that data, the SpikeGLX data and the Phi data, to NWB. Okay, and the data set, the data pops up in this location here. So we have a single session data folder and a multi-session data folder where there's multiple mice and multiple sessions per mouse. So we'll first work with the single session data, and we'll keep this window open because it'll be easier in a second. Okay, and this is all described here, and we'll go there. So I'm going to actually shrink this window a little bit so that you can see some of what's going on. And I'm going to shrink the zoom so you don't need to see my face. Okay, so very first thing we're going to do is to convert the single session. Go to convert and click create a new conversion pipeline. And we're going to give our project, our pipeline, a name. We're just going to call it single session workflow here. And then we can customize where we want our resulting NWB files to be generated into. So you can click this to select a directory, or you can drag a directory in here. But for now, we're just going to leave it as the default, which is going to be under my home directory /NWB guide /conversions. All right, hit next. And this is all described also in the tutorial. So I'll just have that following along as a reference over here. Okay, so next, we're going to specify kind of the type of workflow and the options that we want to be made available to us. And the first thing to do is to specify what time zone is our data in. All data in NWB all time in NWB files are locked to a particular session start time. And so we need to know, when was this data collected and what time zone. By default, this is going to be your current time zone, but you can change this if you know that the data was generated in a different place. For now, let's just leave that. We're going to do a single session conversion. So we'll leave this question, will this pipeline be run on multiple sessions, as no. We'll enter a subject ID. Let's do sub1 and a session ID for this example data set. And for now, we are not going to publish data on Dandy. We'll cover that later. And we're not going to customize the low-level data storage options. Hit next. Okay. Next, we're going to specify the formats that we are going to be converting, and that would be SpikeGLX and Phi. So click Add Format. Here you'll see a list of all the different data formats that are supported, and you can search for any one that is supported. So we're going to look at SpikeGLX. There's two options here. One is the converter and one is the recording. For this tutorial, we're going to focus only on the recording one. The converter is useful if you want to convert many different files generated from SpikeGLX all into an NWB file. But for now, we're just going to focus on one particular recording. So click that. We're also going to look at select the phi sorting. We'll click that. And you can, and when you run this for your own data, you can select your own data and delete and add as needed. Hit next. All right, so now we need to specify where exactly is the data that is from SpikeGLX and from phi. And we'll do phi first. We specify the folder path to all of those phi files. And my Finder, I still have that open. So you can click the box here to navigate to the directory, or since I have it open here, I'm going to go in my single session data folder, drag the phi folder over here. And it shows up green, which means everything is good. The folder contents are what are expected, what Guide expects for doing a conversion from phi to NWB. Then for SpikeGLX recording, we need to pass either the ap.bin file or lf.bin file from the SpikeGLX data, and it will convert just that file. So we'll go to the single session data folder, go to SpikeGLX. We're just going to open that one, open the, and drill down into the individual files. And I will drag the session1 ap.bin folder or file over to this box here. And that shows up green, and we're good to go. We'll hit next there. NWB Guide will extract any metadata that is stored within these files, which can be more or less depending on the formats. And then you can customize those metadata at this level here. So first we're going to go, so I'm going to close this, drag this up, and we're at this section now. We're going to expand, click general metadata to expand it. You can see the session start time has been extracted from the file. The file was generated at 10:11 today. And we can enter any additional metadata that will be useful for anyone else exploring or reusing this data. So for example, where was this data collected, the institution? I will put Lawrence Berkeley National Lab. And lab, the NWB lab, sure. There are certain fields that have yellow text below it, and that suggests that in these fields are recommended fields. And if you do not provide them now, you'll get a warning later saying that these fields are recommended for upload to Dandy and for general good best practices of generating NWB files and sharing data. We'll leave a couple of these empty to showcase that later. But I'll just put an experimenter. Let's put my name. Great. Okay, lots of fields where you can specify additional metadata about the experiments. Then you can go to subject and specify the sex, the species, the age, and other metadata about the experimental subject. You can see that these fields have asterisks and are currently marked in red, meaning that they are required. For this test data, we're going to say that this was a male mouse that was 30 weeks old at the time of data collection. And to specify 30 weeks, we follow the ISO 8601 duration format, where we start with a P, and then a number, and then a letter specifying days, weeks, years, etc. We can also add a description for this subject that may be relevant for your experiment, or a genotype, or strain, or weight. The more information that you provide, the more useful this data will be to someone else looking at it, or even yourself looking at it down the road. You can also click on ECEphys to expand this section, and you'll see all of the metadata about the devices, the electrode groups, the probes, and the electrodes that were extracted from the SpikeGLX data, as well as from the PHI data. Okay, we're going to leave these. You can edit these some of these values, but for now, for this tutorial, we're just going to leave them be. Hit next to move to the next page. Hitting next will run a preview conversion on a small subset of the data that you provided. So let's say your data is 100 gigabytes to start and like 3-hour-long recording. This preview will only consist of a very small time window from the beginning of the recording. Next, you'll see the inspector reports. So NWB Inspector is our validator tool to check whether your file, your NWB file, complies with the NWB best practices. And it will raise any errors that says that your file is invalid, or warnings that's where there are potential errors, or there's metadata that is missing that would be useful for someone else trying to understand the data. So those errors will be shown in red and the warnings will be in yellow. Okay, so we see these two warnings here. We could go back and address those warnings. So let's add an experiment description. My awesome experiments. Hit next. So the warning goes away. If we hit next, it'll run a new preview conversion on the data. And we should see that that warning goes away from the inspector. Okay, then we hit next. Now we will guide integrates with a tool called NeuroSift. And NeuroSift NeuroSift allows you to interactively explore your NWB file. In this case, this would be your preview file. You can see all of the top level metadata on the left here. You can see, my name is the experimenter, the lab, the institution, the description, the session start time is here. You can also expand these headers here to view the raw data itself. So under acquisition, we have an electrical series, and this comes from SpikeGLX. This is the raw acquired voltage traces. And this will show one channel by default. You can also hit this expand button to go full screen. And you can, let's say we want to see the 20 channels at a time. And this is all fake data, but you can kind of, you can use your scroll to move up or down and view different selections of channels. Yeah. Close that. You can also view the units in your data. This fake data has 50 units, and look at raster plots and other properties. Okay, let's full screen this. Okay, and this allows you to verify that the data has been converted correctly, that the metadata has been loaded in correctly, and the data itself are in the right dimensions and generally look the way that you expect. We can hit, if everything is good, you can hit run conversion, and this will run the full conversion, run a conversion of your full data to NWB. And this, depending on how long your recording was or how large your recording was, this could take anywhere from minutes or seconds to minutes to hours. Once that's done, you'll get to this page, conversion review. It says, hooray, your data was successfully converted. You can click this to open the file at its location on your file navigator. And you can move this file around. If there are other data types that Guide that you collected, but Guide does not support yet for conversion, automatic conversion, you can manually adjust this NWB file to ensure it contains all the necessary data. So you can append data like a trials table to the NWB file using PyNWB or MatNWB. And you can follow the examples in the PyNWB and MatNWB documentation to do those. We will also be covering or walking you through that process of using PyNWB and MatNWB for behavior data, for trials data, for extracellular electrophysiology data, intracellular electrophysiology data, and optical physiology data tomorrow. Yeah, and if you have extension data types, you can also use PyNWB and MatNWB to add those data types now that you've created the base file with your base metadata. Okay, so that concludes the single session example. You can hit exit pipeline and you'll come back to this page. You can go back into the pipeline, edit the values and do a new conversion if you'd like. Or start a new one, a separate one. So if you go back to the tutorials, we had gone through all of this, the single session workflow. And at the bottom here is link to the next one, multiple sessions. There we go. All right, so let's imagine you've already run multiple sessions and you want to convert all of those sessions to NWB in a batch conversion. And you don't want to enter the same metadata of your name and the institution and the keywords for every session that you've done. You also don't want to enter that your 5 subjects are all mice. You just want to do that once. We have a solution for you. So we're going to first create a new conversion pipeline. We're going to call this the multiple sessions workflow. We'll keep the conversion output the same. Hit next. All right, and we'll keep the time zone as is, and we'll switch this. Will this pipeline be run in multiple sessions to yes? This creates a new question. Would you like to locate the source data programmatically? If your data are organized, your data for all of your sessions and all of your subjects are organized in a consistent manner on your file system, then I highly recommend that you click yes here, because it's highly likely that you will be able to programmatically tell Guide how to find all of your source data. And now it asks, where is your data located? We'll go back to our test data here, and you'll see that all of our data are in this multi-session data set folder. You might have one for your own data that is similarly structured, and we'll drag that base folder into this box here. Okay, and again, we're not going to publish on Dandy for now and not customize any low-level storage options. Next, we're going to, again, select Spike GLX and Phi as we did before. Spike GLX recording, not converter. And move on. Okay, so unlike the single session workflow, the multi-session workflow with automatic or programmatic detection of finding the files takes you to this page where you have already specified the base directory for all of your data, and you can specify a format string to tell Guide where what is the programmatic path to get to each file, each file folder. Specifying this manually can be a little tedious. So we have a tool here called autocomplete. So click autocomplete. And now you provide one example file folder and specify what are the values within that path that we can then extract and use to generalize to find all of your file folders for all of your sessions and subjects. So let's pick mouse 1, session 2, and all of the file data are in this folder here. You can see, and we're going to drag that over to here, the example folder box. And now we need to say, what is the subject ID in this path? It is mouse 1. What is the session ID? It is session 2 with capital S. And if you hit submit, the format string is generated for you and basically says, the way that your data are organized is subject ID first, then subject ID underscore session ID, then subject ID underscore session ID underscore phi. And using this format string, Guide has found 4 file folders, all 4 of them, in our base directory. And that's great. Happy with that. We did not have to do, drag 4 folders in manually, one at a time. And this would be even more tedious if you had 100 sessions across all of your animals. For your SpikeGLX data, we'll do something similar. We'll hit autocomplete, and we're going to drag the ap.bin file from, let's say, mouse 1 session 2 again over here. And we'll say that the subject ID is mouse 1. The session ID is session 2. Submit. And again, we verify that Guide has found all 4 ap.bin files from SpikeGLX in these various paths. So great. Hit next to continue. Because we're working with multiple subjects across multiple sessions now, we have the opportunity to specify metadata at the subject level. So any metadata that is common to all, common across sessions for each individual subject. So mouse 2, let's say is a male mouse. And let's say at the time of this experiment, it was 29 weeks old. And let's say mouse 1 is a female mouse that was 30 weeks old. And so these metadata values will apply to all of the sessions for that individual mouse. And then we can hit next. Next here, we have the opportunity to customize any values extracted, or customize the data that's extracted from the individual files and where are they located in case you need to override any fields. So here we have the Session Explorer. This is new for our multi-session workflow. Session Explorer is on the left. We can select mouse 2 session 2, or mouse 2 session 1, or mouse 1 session 1, mouse 1 session 2, and expand one of these, and we can see that the folder for phi is where we expect it to be. We can customize whether we want to exclude any cluster groups. For SpikeGLX, there's no other additional options, and you can click through these and verify that the files and folders are where we expect them to be. Okay, now we can hit next. And like in the single session workflow, we can specify the general metadata for each subject and session. And if you open the subject window here, you'll see that the sex and species and age have already been filled out based on the table that we entered data in earlier. So mouse 2 already has male mouse at 29 weeks for both session 1 and session 2, and mouse 1 should show a female mouse at 30 weeks old. Okay, but for general metadata, these fields are currently empty, and we could do this, enter the institution one at a time, let's say Lawrence Berkeley National Lab here, but we would have to do it for all 4 times. Now, to get around that, we have and specify the general metadata that is common to all subjects and sessions. You can click the Edit Default Metadata button here, and this allows you to specify those fields. So institution will be Lawrence Berkeley National Lab. Add item, my name is Ryan. We'll submit that. And let's just, my awesome experiments, sure. We'll submit that. And now those fields are automatically applied to all subjects and sessions under general metadata. So you only have to do it once. Once you've edited, set the default metadata, you can also go in and change, override the values for individual sessions. So let's say under experimenter, for this particular session, mouse 2 session 2, Steph Prince was also helped me collect the data. So that will only affect this session. We hit next. And now, just like before, we are running a preview conversion, but now a preview conversion on all 4 sessions. And we're going to run the inspector on all 4 sessions as well. And we'll see there's a few warnings that appear. And we can go back and address those. And look and use the Session Explorer here to explore the warnings for individual files or individual sessions. You can also use NeuroSift in much the same way as before, but this time you have the session selector on the left here to select the different data. And you'll see for mouse 2, session 2, that the experimenter correctly shows both me and Steph as the experimenter, but session 1 is just me. This window is a little small, so you can go click the full screen to really expand NeuroSift and browse the data. Okie doke. And then once you hit run conversion, it will batch convert all 4 sessions to NWB. So we should see that here. And as I mentioned before, this can take some time depending on how large your files are and how many you have. Great. Data was successfully converted. Here are the links to the individual files. You can see them, they've been placed according to the subject. So session 1 and session 2 are both under submouse 1, and session 2 submouse 2 session 1 and session 2 are both under the folder submouse 2. And like before, you can go into PyNWB or MatNWB and open these files and append any additional data to them. Great. So now we'll exit the pipeline. And we've completed that tutorial. And I've showed you how to convert data from a single session to NWB, as well as multiple sessions from multiple subjects to NWB. Are there any questions before we move on to uploading data to Dandy? Okay, we'll move on. So on the tutorials page, you can go, we've gone through this whole page already, and we'll click dataset publication to move on to the next. Now this, as I mentioned earlier, this tutorial requires that you have an account on the Dandy development server. If you don't have an account yet, you can follow along or and do this section later if you'd like. Okay, so we're going to go to let's say let's do the single session workflow. We will resume the conversion and we'll navigate back to the pipeline workflow panel here, page here. And we're going to say that we actually do want to publish data on Dandy. Select yes. And we can go back to the conversion. And I think if this works as I expect, we hit next, it will take us to the Dandy upload page. Great. Okay, so for this to work, we need an account on the development server and to pass, to give NWB Guide our API key. And I know that this video will be recorded and uploaded to Zoom. So I'm going to put my API key and then after this video, I'm going to reset it. So to get your API key, go to GUI-staging.dandyarchive.org. And if you sign in and click your account icon here, your API key will be this long hexadecimal string here. I'll hit copy it to keyboard, and then click API keys here, and I will pass in my API key there. If you want to upload directly to the main Dandy archive server, then you would do the same steps, but use but go to the main dandyarchive.org page and get the API key from that page instead. I'll hit submit. Great. And now we can select which Dandy set we would like to upload to. If you have an existing Dandy set that you've created on Dandy and you want to add files to, you can search for that here. Or you can create a new one from scratch by clicking the Create New Dandy Set button. And we're going to follow those steps here. So let's say this would be NWB Guide Data Conversion Workshop Test. The staging website of Dandy is used primarily for development and testing. Don't put data on there that you don't have anywhere else. Like, it will get cleaned out regularly. And putting data there does not count as publishing it. And, you need to put it onto the main archive for that. We're going to select the development server because that's the only one that we've specified the API keys for. We need to provide a description, test, upload during data conversion workshop. Just put anything here. And then Dandy requires that you provide a license for your Dandy set. There's two options, CC BY or CC 0. You can review the individual licenses on the Dandy website. I believe the Dandy handbook has a guide for what these mean and why you would want one or the other. Click Create. And this will create a new Dandy set. This one is number 214528. And I can set any additional settings if I'd like, but let's leave that for now. And then hit upload. And this should upload the one NWB file from the single session workflow to the Dandy set 214528. And it is successful. And so once you get to this page, you'll know that it has uploaded, and you can click this link to launch a website, your web browser, where you have your new Dandy set with the name, your name, and there's one file in it. You can go to files and see that the file is there, and you can download the file. You can open it with tools here, and make sure that everything worked. Okay, and you can also add additional metadata, and we encourage that if you're publishing this data for real on the main server. Okay, and exit and to save, and that's it. that is NWB Guide in a nutshell. Do you have any questions?